Hi everyone, this is the last lecture of topic 2. In this lecture, we are going to learn about the life cycle of a cell. A cell has two major phases in its life cycle. The first phase where it is growing and preparing for the cell division. That phase is known as interphase. The second phase is where it is dividing. The name of this cell division is mitosis. So by mitosis, the cell replicates itself. From one mother cell, two daughter cells are produced. So the cell life cycle has interphase followed by mitosis or cell division. In mitosis, the chromosomes present in the nucleus of the cell are divided equally into two, and this happens in four subphases: prophase, metaphase, anaphase, and telophase. And this is followed by the division of the cytoplasm, which is cytokinesis. And cytokinesis may overlap with the last two subphases of mitosis, that means anaphase and telophase. An easy way to remember all the phases in the life cycle of a cell is to remember this mnemonic IPMAT. I stands for interphase, P for prophase, metaphase, anaphase, and then telophase. This figure illustrates the cell life cycle. Everything that you see in blue is interphase stage of the cell cycle, while mitosis is shown by red color. You can see that the interphase takes way longer than the prophase. It would take about 20-21 hours for a cell to complete interphase, while the actual cell division or mitosis is done within 3 hours. And then the mitosis would be followed by cytokinesis, which is division of cytoplasm, and then the cell goes back to interphase. Let's start with interphase. In the interphase stage, the cell is preparing for cell division. So one cell has to divide into two. What the cell needs to do is double its contents. That means it has to duplicate everything that is present inside the cell. And interphase consists of three subphases: G1, S, and G2. G1 stands for gap 1 phase, S stands for synthesis phase, and G2 stands for gap 2 phase. In gap 1 or G1 stage, mainly duplication of organelles occurs. That means all the organelles double in number. So let's say if a cell originally had 5 mitochondria, right before the cell undergoes mitosis, it would double the number of mitochondria to 10. In S phase, S is for synthesis. DNA replication occurs, DNA synthesis. That means the cell makes or doubles the amount of DNA that it has. In the gap 2 phase, protein synthesis occurs. That means doubling of the amount of protein. This figure shows DNA replication. That means how the DNA makes a duplicate copy of itself, and we are not going into the details of DNA replication here. The first stage of mitosis is prophase. So in prophase, the nuclear membrane, it starts disappearing. It breaks down. There are a pair of centrioles which start moving in opposite directions. So they move to opposite poles, that means two opposite sides of the cells, and later on they would start giving out mitotic spindle fibers, which are made up of microtubules. The chromatin, which was present in the nucleus as long, thin thread-like structures, they now start condensing. They get shorter and thicker, and they now appear to be double-stranded chromosomes with two sister chromatids. The second phase of mitosis is metaphase. And metaphase, the most important feature is that all the chromosomes will line up in the center of the cell, and this central part of the cell where the chromosomes are all arranged in a linear fashion is known as the metaphase or the equatorial plate. Metaphase is followed by anaphase. In anaphase, the two sister chromatids of each chromosome are pulled apart by the shrinking microtubules, that means the spindle fibers, and the two sister chromatids start moving towards the opposite poles of the cell, and when they are doing so, they look like V-shaped chromosomal elements. And the cell in general looks long, so it begins to elongate. Anaphase is followed by telophase. Telophase is the opposite of prophase. In telophase, the nuclear membrane starts to reappear. The chromosome, which was short and thick, it starts becoming long and thin again and becomes chromatin. The spindle fibers break down and they disappear. After the chromosomes and the nucleus has divided into two, it's time for the cytoplasm to divide. This division of cytoplasm is known as cytokinesis, and it usually starts during anaphase and it's completed by telophase. In this, initially a furrow forms right in the middle of the cell. This is known as a cleavage furrow, and it gets deeper and deeper, and then it pinches or splits the cell apart into two daughter cells. This figure illustrates all the events of mitosis starting from interphase up to cytokinesis. This is another figure showing the mitotic phases. This slide and the next two slides after this show diagram as well as photographs of cells undergoing mitosis. This slide very clearly shows the metaphase plate or the equatorial plate, and look at the V-shaped chromosomes in anaphase. This slide shows the end of cell division. It shows telophase and cytokinesis.